with our next disruptive dialogue discussion. I'm Keith Matthews. I'm from Sidley, Austin. We're one of the sponsors here. We are a regular sponsor, a yearly sponsor of the Ag Innovation Showcase. It's a wonderful venue. We are happy to be here every year. I'm always happy to come back here every year. I actually grew up in the St. Louis area, so it's a wonderful opportunity for me to come back to what initially at least was home for me. I'm also very pleased to be in a position where I'm following the panel that went on earlier this morning, as well as the company presentations, and also introducing this particular disruptive dialogue. As I mentioned yesterday during the special content session, legal counsel related to and work related to biopesticides has been a major focus of my career for over 15 years. And this is something that is actually very important to me. Biopesticides, biologics, what's going on, what has been going on with them is very important to me. It has been a major focus of my career. And I'll note that EPA got out ahead of the curve over 20 years ago when it established a separate standalone division to regulate and register reduced-risk biopesticides and to promote integrated pest management, or IPM. This is something that at the time there wasn't a lot of thinking that was going on around this, but EPA recognized that this was a future of pest control, or it was going to be a major focus, a major component of the future of pest control. So it established a division, which I eventually came to head for about 4 years, that would focus on a reduced burden, providing a reduced burden to the regulatory process for registering biopesticides. Now, having said that it's a reduced burden, as I discussed yesterday in a special content session, registering a biopesticide is timely, it takes time, and it's also expensive. But it is much less expensive and much less timely. It takes much less time. I shouldn't say much less timely. It takes much less time than does registering a conventional pesticide. Now, interestingly enough, however, one of the things that the agency does not look at when it's looking at the question of registering a biopesticide is a question of efficacy. And so it's very interesting that, and very topical, that the disruptive dialogue we're having here this morning is going to talk about a major challenge to the integration of solutions is a testing for increased efficacy, because the agency leaves efficacy to the private sector. It leaves it to the market to figure that out. And so that's what our panel is going to do today. And so I'll bring to the podium Jim Budzinski, who's going to chair and moderate this session. And Jim asked me to note for everyone that with respect to questions, you can either send questions to the hashtag on Twitter or to his email. And so with that, I leave it to you, Jim.
Good morning. Are you ready to talk biologicals? You know, Venus and Mars, good and bad. You know, that's kind of the dichotomy we have, right, as an industry, is everything new coming in is kind of the new stuff, and all that stuff that was there, that's the bad stuff. And then all of a sudden it reaches a point where people say, hey, you know what? A little bit of both of these might be a pretty good deal. And that's really what we're going to try to talk about for the next hour, which is how do we integrate these two things moving forward? So you're all familiar with the Twitter thing. Here's my email if you're not a Twitter guy and you want to send a question, because what we're going to do is spend about 30 minutes having some people who know a lot more than I do about biopesticides and biologicals talk, and then we're going to kind of open it up and try to spend at least 20 to 30 minutes at the end taking your questions about this area and how this is all going to come together. Now, you all probably, by this point, have heard about Ag 3.0, which was what I spoke about here 2 years ago at the closing keynote. And Ag 3.0, simply put, was how is Ag changing as we go from the 50 years of consolidation to kind of a new era where resources are not cheap. And for 50 years, water was cheap, and chemicals were cheap, and fertilizers were cheap, and seeds were cheap. And we used a lot of them, sometimes too much. And Ag 3.0 is kind of what comes next when you got the farm about as big as you can get it, and the consolidation is taking place, and we're talking about what's next. So one of the categories we talked about in that was what happens in the weed control space as we move forward. And it's really this movement from what I'll call a reactive and curative to a proactive preventative. And you'll note synthetic and natural solutions coexist. And that's really what the next hour is about, is how do these two coexist? So here's the playing field, right? The phyllosphere above ground, the rhizosphere below ground. And the bottom line is we're learning a lot every year about how plant growth can be separated into these two. And for those of you who were in the presentation earlier, on mycorrhizal, there are all these communities that have been there for, in some cases, millions of years, right? That were doing a lot of things we didn't know about. They had a role in plant nutrient availability, plant regulation, disease suppression, interaction with genetics. So it's really, how does this play? Now, as a guy with a degree in soil chemistry, I guess I have a particularly keen interest in this. If you look above ground, the world's about crop and weed characteristics, physical, chemical, and biological, spray solution, spray dynamics, and then drift and volatilization. Think of those as the above ground things we worry about. And particularly of importance to this discussion is the one at the very top up there, I can do this right, which is what happens in the tank and how do we integrate things from a formulation standpoint and a performance standpoint. But where it really gets interesting is below ground. Because below ground is where these three spheres that they taught us about in school, right? Physical, biological, and chemical. And for most of our careers, they said, well, there's a lot of stuff going on there, and there's organic matter, and there's 20,000 different things in there, and don't worry, it's all good. Right? That's what we did for like 40 years. And now we're starting to untangle the spaghetti, right? And you've got things like, what's the porosity and infiltration and holding capacity look like? What's the soil solution look like, which is really a chemical medium? And what's the water-based game in the middle biological paradigm, which is really where we're at today. And tomorrow when we do the closing keynote, we're going to talk about what are the key opportunity areas. But today what we want to talk is, how do you integrate these into a program moving forward? Now, my own bias is what I call integrated agronomic management, which really is focusing on the ABCs, agronomy, biology, and chemistry. And for most of our agronomic careers, the agronomy guys were the equipment guys, right? And the chemical guys were often fertilizer guys, right? And over here were the seeds and the biology guys, and they all kind of had their own piece of the pie. And we'd mix it all together at the end and hope it worked. And now we're realizing, you know what, the only way this is going to work is if we actually mix them together at the beginning and figure out how they ought to work together. Most of our work is empirical, not mechanistic, which is, hey, I tried this and it works. I really have no idea why or how. And as we get a little further down this road, we're going to start figuring out, hey, that did this because of this. That's when we really start getting traction with some of the things we're talking about. The interactions will be better understood in agronomic. So, with that as kind of a setting the stage, we have a great panel today talking about key questions in this area. And what I've done is ask them to come up and to a greater or lesser extent focus on these 5 or 6 questions here. Why should biologics be added to the crop protection toolbox? How can we integrate them with chemical solutions? What challenges do we face? What's your company's approach to integration? What are the benefits of hybrid solutions? And particularly, what does it mean for formulations and optimization? And importantly for those of you who are more entrepreneurs in this area, what kind of advice would these gentlemen give you in terms of, as an entrepreneur developing a technology in this vertical, what things do you need to be concerned about and thinking about? So we're going to start out with this in this order. Mike with New Leaf, Mike McFatrish with New Leaf, who has a very unique background in this area, and we're going to let him tell you a little bit more about it. We got Brad Griffith with Monsanto Novozymes, Jean-Philippe Albert with Syngenta, and Joe Smith with Bioconsortia. And each of them will have a unique perspective, some from the perspective of big companies trying to integrate that have a pretty good portfolio of products already, and some from the perspective of companies with new technologies they're developing that are trying to fit into what's going on. Because at the end of the day, it's both of those perspectives, I think, that'll be the most useful. So remember the Twitter, and remember if you would like to send an email with a question, and when we get done here, we'll open it up and spend about a half hour talking. So first, we'll have Mike come up and talk a little bit about New Leaf's perspective on all this.
Thanks, Jim. Well, I appreciate having the opportunity to be here today. It's a pretty exciting area for me, and certainly, believe it or not, even though I appear incredibly young, I've been doing this for about 21 years. In that respect, I guess, in terms of the unique background, I come from a technical side where I started out in varietal and hybrid plant breeding and gravitated more into the commercial side. And I found it interesting when I moved. The interesting part of being able to use chemistry in combination with biologicals, and that, in fact, is the opportunity that I think we have. And I think that when you talk about the emerging ecosystem of the biological markets, I think, as Jim pointed out, one of the key elements is that we're moving from curative, SDHI chemistry or strobilins or carboxamides, to a preventative, a much more plant-based, a much more unique and tailored fit. Indeed, what we like to talk about at New Leaf in terms of prescriptive biologics. And in that respect, I think we're working in a space, New Leaf, with methylotrophic bacteria, pink pigmented facultative methylotrophic bacteria to be precise, and for my commercial plug for today. To be able to understand and be able to work in a much more broader sense, not only as standalone products, but also with in combination with chemistry. And so I think understanding the microbial host plant, being able to understand the genetics, being able to create collaborative relationships between the plant, between chemistry, and between those microbes is a pretty important part of how we view what we're trying to achieve. And if you look at in terms of yield response and those sorts of things, in terms of the growth, obviously curative has added a lot. The utilization of fertilizers, herbicides, and of course foliar applied fungicides, whether it's in furrow or on the plant or on the seed, which is a lot of where I've spent my time, has brought us to an incredibly high level of production. And of course, that well-worn phrase, doubling food production by 2050, is indeed what we're after, but not just doubling it, being able to do that in a manner that is, and forgive me, sustainable. And that's where I think we fit. Can we do more with less? And in that respect, I think the managing abiotic stress and complementing genetics, complementing the plant genetics, not only of the crop, but specifically of a hybrid or variety, allows us the opportunity to leverage the technologies that we currently have and be able to build upon that platform and be able to move forward in a much more rational and forgive me, sustainable manner. So with regard to the questions, I think that, I think enhanced performance, being able to do more with less is a critical component, a critical element of what we bring to the table in terms of our toolbox. And I'm not just talking about New Leaf, I'm talking about us that participate in that biological sector. I think being able to do that, doing more with less, and being able to prolong the life of certain chemistries in an arena where obviously one of the aspects that I feel very strongly about is we're not talking about elimination of chemicals. I mean, clearly that is part of what we have to manage. But can we do that in a fashion that allows us to preserve the technologies we have, preserve the environment in which that we participate, not only in terms of the physical environment, but the cultural, political environment as well. Certainly our colleagues from Europe can appreciate the implications associated with the stresses that are put on carboxamides and, of course, the neonicotinoid situation. So for me, it's sustainable, being able to enhance that performance. So that's why I think biologics can be added to the portfolio. And so how do you know, in terms of the integration with chemical solutions, well, this is pretty much a lay down hand, compatibility. So what's that compatibility look like, and being able to determine what that compatibility is with not only those biologicals that you might be working with, but obviously the chemistry as well, and being able to understand what those doses are with regard to the application rates. Because a great farmer phrase that I love a lot is, you know, in any kind of crop protection tool, a little's good, a lot's better. Well, that's not true. Well, perhaps in some instances. But in terms of being able to utilize the technology and understanding and being able to understand what those doses are. So I think compatibility and understanding dose and understanding complement in that compatibility. So what's our approach to that integration? Our approach is to look at our methylotrophic bacteria in a manner that allows us to look at it as a standalone strain, to look at it as combinatorial with other biologicals, and to be able to look at it equally important as how can we complement traditional crop protection, traditional AI. And that's an area that is really pretty exciting to me, because I think we're doing it in a manner that is very precise, very focused, and on key elements, understanding what those key AIs are and how they contribute. So what are the potential benefits? And what are the potential benefits of these combinations? Doing more with less. Being able to extend that window of crop protection. Being able to potentially preserve tools that we currently have. And being able to perhaps provide substantially similar performance with slightly lower dose rates of existing technology. But the part is being able to partner. And certainly the genetics of the plant and the genetics of the crop, more broadly, are an important part of understanding of how you extend that window of protection and how you complement in a fashion that you're able to provide a product offer, a technology, that achieves that substantially similar performance or enhanced performance with either a lower or substantially similar rate of application. So that's the benefits as I see them and as we see them. In terms of formulations, how can combined formulations be developed and optimized? For us, it's taking an approach that is a tiered approach. And that tiered approach is understanding through exhaustive and focused research where our technology, where our microbials, what they do, and where that performance is coming from. And then being able to take that to the field and be able to look in how it combines, so that we have an understanding early on of exactly what we contribute and what it might be partnered with. So it's a tiered approach in terms of formulation development. Of course, as I said at the onset, it's being able to look at that in terms of compatibility and understanding what that fit is. So, in terms of, we are a startup, relatively new, but we've come a long way, and I think we're right on the cusp of delivering technology, which we hope to have in the market next year. So for our perspective, what are those things that someone coming to this? I think from our perspective, the thing that drives us is relevance and understanding where that performance comes from. And by relevance, I mean not only the relevance in terms of what that performance is in terms of gain or disease reaction, but making sure that we focus on things that are important. I have a friend and former colleague that in the biotechnology world loves the story about having given advice to someone who was developing a poinsettia. And they thought that, well, we can have various colors of poinsettias, and we'll be able to charge $200, $300, $400, $500 for those plants. Well, guess what? Maybe 3 or 4 people might like that. But I mean, I don't know that at Christmas time I'd want like a blue poinsettia. And the same thing with understanding the market in general, and being able to understand that relevance, and being able to demonstrate the performance enhancement that you deliver. And I think that that's really, really important, and also understanding where that performance comes from. So I appreciate having the opportunity to share my observations with you, and look forward to a very spicy question and answer session. Thank you.
Good. So next we have Brad Griffith with Monsanto Novozymes, and he's going to give us a little bit of their perspective of how they have been trying to integrate as they look at the two pieces of their business.
Thank you very much, Jim, and thanks, Mike. Good talk. I've not yet succumbed to readers, but when I do, you're going to be my primary coach on how you flip them up and automatically bring them down. That is a skill. That is a skill. Well, I'd like to start first by thanking the Danforth Plant Science Center and also the folks at Sidley for inviting and affording us at the panel this opportunity to address you on something that's kind of small, but could be the next big thing in agriculture. And I'm joined today by a number of colleagues who made the trip from across the street to the Danforth Center. And I'm also joined by colleagues from Novozymes who made the trip from Canada in some cases and from Denmark and others. And I want to thank you all for coming and joining us today. As we are out and about in a network session, I would certainly invite you to pull any of us aside and ask us a bit about our BioAg Alliance. We'll tell you more about it today. My name is Brad Griffith. I've been with Monsanto for 14 years. I've been in the life sciences for 24. The first 16 years of my career I spent in the pharmaceutical industry. And you think the transition from pharmaceutical industry to agriculture, at first it's a bit angst-provoking. I'm not going to lie. But what has really blown me away over the past 8 years is just the amount of technology that has hit agriculture and have hit our farmer producers and our retailers, our distribution partners. It is an immense amount of technology. I mean, it's humbling when you sit back and you think about genetically modified crops and biotechnology. We're talking about technology that's 20 years old, 1995, it's 2015. So I'm old enough to have lost my hair and gained weight. During that hair loss and weight gain time, I've had the ability to send twins to college. They're 20, they're sophomores. So when you think about genetically modified crops and biotechnology, we're talking about a sophomore in college. When I look at this technologies like what Mike described, we're very early in a phase, and we're learning to walk and talk. And they're saying words like penicillium bali and Bacillus amplifications, and words like what Mike said. So we're often asked at Monsanto, what did you see about the category, and what was it about ag microbials that made now the time to enter it, and what were some of the strategic rationale for why you did it? There we go. We see ag microbials as an input, a pivotal input in helping farmers maximize yields and mitigate risk. All right. And when you look at this particular chart, we estimate that today the market is valued at about $3 billion inclusive of BT technology. When you exclude the BT technology, we would estimate it to be closer to about $2 billion. When you also look to the right-hand side of the slide, you see that the fruit and veg space is kind of the primary user today, and there's some fairly significant row crops with insignificant penetration of ag microbials, certainly opportunistic for this category. When you come to the bottom and you contemplate the crop nutrition business valued today globally at about $185 billion, macro and micronutrients, and you think about the crop protection business today valued globally at about $65 to $67 billion, And you think about this $1.8 to $2 billion that we have in microbials, imagine this. Can we live in, and I believe we can, but can we live in a world where we can reduce crop nutrition maybe by 5, 10, 15%? Can we complement current synthetic chemistries for the control of disease and pests to mitigate risk on a farmer's crop? And if so, what do those values have? You know, I would argue when you look at a pie as big as 185 billion, you look at a pie as big as 65 to 67 billion, and you look at the opportunity that stands before us to innovate in this category and to provide farmers with those sustainable solutions that Mike talked about, it gets pretty exciting. You know, when you do look at some of the global opportunities and the magnitude of the row crops, you know, if you focus on corn, 425 million corn acres globally, rice, 400 million acres globally, wheat, 540 million acres globally, and look at the microbial penetration in those crops today. There is a significant opportunity for people on this panel and for people in this room to bring innovation to this category quickly. We can bring innovation to this category quickly. You know, and I think back a few years ago as to how Monsanto got involved in this space. Our CEO, Hugh Grant, interacted with the Novozymes CEO, Peter Hoek Nielsen, at Davos. And the discussion was, this appears to be a pretty significant opportunity. Is there a manner in which we can go about this together and try to solve this more thoughtfully, leverage resources and capabilities of your business, your enzyme expertise, leverage resources and capabilities from our business, Monsanto, Global Germplasm and Testing Network? It seemed to make a lot of sense. And that's exactly what we've done. Wow, check out that build. So when you think about bringing together two organizations and leveraging synergies, but also respecting and leveraging complementary capabilities, that's precisely what we've tried to do. And that's the way that this alliance was set up. So, you know, sharing with you in a very transparent way, our Novozymes colleagues have expertise in small-scale fermentation as we're building these discovery microbes to get out and test in the field. An example, you know, our folks in RTP from Novozymes were able to build out over 2,000 discovery microbes for us to treat seeds in February and plant this last campaign. 2,000. They started in the September-August timeframe, and they were successful. So it was a daunting task, and kudos to them for that. But they clearly have large-scale, small-scale fermentation expertise, process optimization in the production of these enzymes and ag microbials. On the Monsanto side, you know, we bring the regulatory submission expertise and capabilities globally. We bring the commercialization, the sales, the marketing capabilities globally. And this field testing piece that I'm sure we'll talk about a bit more, we're really trying to drive a bit of a paradigm shift here by going to what we call field first. And when you go to field first, you know, we have a very significant number of plots out today, probably more significant than have ever been tested in the history of this category today. And it gets bigger. And we're being more thoughtful with our colleagues from Novozymes about the genus and species and placement of these different microbes, because today the science has afforded us that ability. Our knowledge of the genome and our ability to map where we see successes and where we see failures help us be more predictive about where we put these microbes in the future. So it's very exciting. Now, as you sit in the audience, you say, okay, great. Good for you, Novozymes. Good for you, Monsanto. What does this mean for me? We purposely allowed 3 different inflection points that we've highlighted here at the bottom. Inflection points where we welcome discovery collaborations early. Both organizations maintain independent but fully alliance-committed discovery campaigns. Our development efforts are split 50-50 between the organizations, and our business contribution is split 50-50 between the organizations. So we welcome the opportunity to bring discovery collaborations in early. We can also in-license products, technologies, or platforms through that development process. And on the out-licensing side, we have technologies that we can certainly talk to you if there's good synergy and fit in your business and organization to explore out-licensing opportunities. Our expectation is that by 2021, 2022, as some of our discovery microbes begin to hit, we may have more microbes than we know what to do with. And I think our approach has been one of empiric. We're going after that broad acre efficacy. And we know that we're going to see some regional targets that hit extremely well and some targets that hit extremely well on crops. We're taking a little bit of a broader approach, at least initially. So I think there's a significant amount of collaboration opportunity that exists between all of us in this room. So just to show you some examples of different types of prospecting opportunities. Of course, microbes, consortia of microbes are interesting, but then in terms of enabling platforms, there are different types of platforms where the Alliance could use some help. And if there are folks sitting in this room or there are people that you know the power of networking, we would certainly appreciate the opportunity to talk to you. It's my understanding that in your bag you would have received something that looks like this. And on this is just a bit of a synopsis about the BioAg Alliance between Novozymes and Monsanto. And then when you open it up, you will find the schematic that we just worked through, but you're also going to see this schematic that talks about benefits that could be conferred to parties in this room as far as maximizing and tapping into innovation together in a collaborative way. So I encourage you to look at this. We've also launched a new website. It's www.monsantobioag.com/thebioagalliance. And right there, you're going to find all of that information as well as contact information to email someone from the Alliance if you have a technology or you have a platform or something that you would like to talk to us about. That's what really gets us excited. As I think about wrapping my time up, Mike talked about 2050 and the challenge of doubling yields in 2050. It's a real challenge. I mean, the statistic that blows me away is the fact that in the next 50 years, we have to produce more food than has been produced on this planet in the last 10,000 years. It's hard to wrap your head around what that really means. And think about the amount of productivity gains and the inputs and how these systems, these disruptive systems, will play a pivotal role. But they will. In 1960, one farmer fed 26 families. Today, that farmer feeds 155 people. How many do you have to feed in 2050 when you add the equivalent of another China, a couple more billion people to the planet? I love the fact that we're breaking ground here on the Danforth Center. To me, it speaks to innovation, it speaks to creativity, it speaks to entrepreneurship, but it speaks to hope. And we're honored to be a part of that hope. I hope you share the vision of our four panelists in this particular category. And I certainly thank you all for your attention and look forward to the Q&A. Thank you.
Excellent. Very good. Jean-Philippe Albert is with Syngenta and actually has quite a similar footprint that, you still have the clicker, or is it up here? So Jean-Philippe has a very similar role looking at a lot of Syngenta's programs, and he's going to share with you a little bit about how they look at some of the same things.
Thank you, Jim. Good morning, everyone. So as you will soon realize, I come from Europe, from France specifically. We'll give you today an overview from probably more the grower and the marketing side of things. Before diving into the topic and breaking everything here, I just want to thank you, Derek Norman from Syngenta Venture, who is giving me the opportunity to be with you, and I consider this as a great privilege. So briefly, my background, I've got an agronomy background. I spent the last 17 years with Syngenta and its legacy company, working in sales, being very close to growers, promoting crop protection product. Then I had several roles in the French headquarters, and then in Basel, which is the global headquarters of Syngenta, where I had the pleasure to work with fungicides, herbicide, various crops, and recently was appointed as head of new technology, so it's maybe a big word, which means that I'm looking after all our assets on the biocontrol side and on crop enhancement, which is another way for us to characterize abiotic stress management. So having said that, as I said, I will take you through the prism of the customer needs in this realm of biological product. Probably very often during my pitch, I will make a clear distinction between biocontrol product from biological origin, which have been designed to control pests, plagues, you name it, and as well the biological product used as biostimulant to improve the plant ability to face abiotic stress or enhance nutrient uptake. So look, what I do propose, I will take the questions one by one. This is the way I've prepared this speech. Why should Biologics be added to the toolbox? And to be provocative, I understood it as to be a disruptive dialogue. Do they need to be integrated? To be very provocative, I would say as long as the growers have efficient seeds, efficient seed treatment, efficient fertilizer, why should they use a biological product? Because they like flowers and birds. You know, what we see very often is that growers, they look at their P&L. As long as they have freedom to operate with the chemistry they have in the toolbox, why should they jump to in the realm of biocontrol product or biostimulant? And in fact, the answer, or at least one of the explanation I propose to you today, is that when some chemicals are limited from a regulatory point of view, or technically limited because of resistance, therefore new tools are needed in the box. And this is where, of course, biological will be new tools in the box. If suddenly, in whatever region of the planet, a local authority decides to ban a given pesticide to preserve, I don't know, groundwater and the like, we need other solutions. And as worldwide the regulatory is becoming more stringent to register new pesticide, biological presents certainly a wonderful opportunity to discover new mode of action. So this is the first driver. Why should grower use biological product and more specifically biocontrol? Very often, in the vast majority of the country I know, biocontrol products are not considered to leave residue in the synthetic meaning of the word. So if you're a winemaker in Napa Valley, for example, you want to export your wine to Switzerland, where I'm working. You may have an interest to use biocontrol product at the end of the season as they won't leave residue in your wine, and therefore you will improve your market access. This is the case in India, for example, which is exporting table grape to Europe and the rest of the world. Second point. The third driver, and you may not be interested with it, is if you are a zealot of the organic farming, you may need biological because it corresponds or it matches with the protocol you have to follow. But let's be lucid about that. This market segment exists and will continue to exist, but that's a niche. And if you are the CEO of a startup, you may think as well, which is the kind of market you want to target. So that's where Biologics should be added to the toolbox. How can they be integrated with chemical solutions? I think that's an easy question, but that's very difficult to answer. So I would propose to answer in two steps. Integration, what does that mean? You're a grower, going back to the customer. You have tools, you use them in the toolbox, you have a crop to protect. Let's think first above the ground, right? Classical crop protection business. You have two solutions. Either you combine your biological product with a conventional, or you alternate with the classical chemistry. So this is what we've done over the last 5 years at Syngenta, looking what would be the best way. I may disappoint you, but there is not a single right answer. It depends of the crop, it depends on the pest you want to control, it depends on the mode of action of the product. Needless to say that this is not with 10 trials that you will crack it. You need to invest, and I think, Brad, you mentioned it, hundreds of trials, sometimes millions, before being able to demonstrate the value of the proof of concept. Why am I insisting on that? Because I know it's different here in the US. In some other area in the world, specifically in Europe, when you want to bring an innovation in the market, you need to demonstrate that it brings value to the customers. You need to demonstrate the efficacy. So if you don't have in your package all the data set which will demonstrate that your technology will bring added value compared to the existing solution, it will be very difficult, to say the very least, if not impossible, to register it. So you have to do this first experimentation. Now, you can as well think about mixing products together. You want to wrap a seed, for example, if we talk about seed care, and you can mix biocontrol product with a conventional fungicide. The same in a spray tank. But let's go back to the basic of science. If you want to mix a bacteria with some copper, for example, copper is a super powerful bactericide. It is used to sanitize a lot of places. So I think you need to do the studies, but very obviously, you may have some inhibition. It's an example we could multiply. So again, you need to test the compatibility of your biological solution with the conventional. Integration very, very often is as simple as that. And again, it will depend on the strategy of your startup. It depends where you want to go. Maybe if I can afford a piece of advice, an easy way would be to dream that you would mix your biological solution with the blockbusters of the major R&D company, and it will fit, it will be okay. I'm afraid it may not be sufficient. So let's be lucid about that. So what are the potential benefits of hybrid solutions? Let's be extremely selfish for companies like the startup of the big companies. The benefit may be to have exposure to a bigger market. We may have as well patent benefit. Let's assume a company like us, we may have an old product which is out of patent. We are threatened by generics from China or whatever. Suddenly, we have the possibility mixing our older asset to a nice biological strategy to get patent protection, to rejuvenate, and lowering the rate as well of our conventional AI to access new market. Or even more, to, for example, face resistance issue, right? So these three elements. So patent protection, this is a good idea to combine. It's really business-driven, sorry. Going back to the grower, you can mix to tackle resistance. You have a product which is not working. You mix it with a biocontrol, suddenly it works better. And the third point as well is if the authority told you, look, you can spray this nasty pesticide once a year or twice a year, but at half the rate. You need to find another balance, and biological product may help you there in this direction. Maybe I will spend a bit more time on the last point. What are the most important things for developers of biology technology? As I want to end on a positive note, I will start with a negative one. From experience, probably the base case you may have in your respective companies to develop a solution. This base case, you have to be ready to see it more difficult. It will take more time, and it will be more expensive than you have already planned. And talking from experience, we see that very often. Do not underestimate the difficulty. And this is not because you have in a petri dish a powerful strain which is able to enhance nutrients uptakes everywhere, that it will work in all the possible directions. I think we had the discussion with Joe Schmidt yesterday. If this strain works in a clay soil, will it work in a soil, which is richer from an organic matter point of view? Will it work when the soil is dry? Wait, you have a lot of parameters. So this is very important point to consider. So now, in terms of important things, first, the proof of concept, you need to understand what you are selling and how it works. In some countries, it may be very easy. You may not have to justify how it works and why it works. Good for you. Now, if you want to make of your startup a bigger company or make it attractive for bigger companies, please make sure you know exactly how it works. Again, talking for experience, we had sometimes some surprises. Point number 2, in terms of production, if you want to go global, to play in the big courtyard, you need to be sure that the product you are proposing may it be a biostimulant, biocontrol, could be produced at scale with a viable as well economic process. Again, from experience, we know that producing at scale is sometimes tricky. Sometimes it's not, but it doesn't make sense from an economic point of view. Right? As we are not working with synthetic compounds, but with living things, we need to consider all the conditions which are needed to make them grow. Right? I think, Brad, you're nodding because as well your experience allows you to. Now, in terms of efficacy, I think I have insisted enough. You need to be sure that you can demonstrate the benefit of your product. If you don't have to, good for you, but the market will have the last word, that's for sure. And if you need to demonstrate the benefit as well, you may design specific protocol. When I scout the websites of the startups in this biological world, I'm very often surprised to see the little attention which is paid to show, the added value. Very often you see a chart with no references, no trial number, no starts. So for big R&D companies like us, sometimes that's not enough, and you need to spend time as well there. Probably just to conclude this word, I think what we've seen recently, after years of investigation in the world of biostimulant and or biocontrol, some new technologies are really able to play neck to neck with conventional chemistries. If you find a way to integrate this solution in the program at the right place, on the right crop against the right pathogen or abiotic stress, you can absolutely compete in the big lead. But again, it takes a lot of rigor, a lot of time, and of course, a lot of hope as well. Thank you very much indeed.
Thank you very much. Joe Schmidt with Bioconsortia is going to close us out here. Hey, Joe, we have like 30 seconds left. No, I'm just kidding. No, we are actually going to take questions after this, so I know we're running a little long, but I think this is a great discussion. So we're going to let Joe go now, and then we'll take questions from the audience.
Okay, first I just want to thank the Ag Innovation Showcase and LARDA for this opportunity to get in front of you and speak about something that I'm extremely passionate about, and that is biological solutions, but more importantly, how those biological solutions can be integrated with the standard agronomic practices in seed, fertilizer, and chemical pesticides. Having worked most of my career in agriculture and in emerging technology companies, and then most recently coming from Bayer Crop Science, where I was Director of Global Business Development for mainly around the biological areas, in licensing and as well as bringing new acquisition opportunities to the company. I think I have a relatively unique perspective on what it takes to be successful when it comes to massive adoption of biological products. So before I dive into how do you integrate those most effectively with chemistry, I want to talk a little bit about what, in addition to the market pull, all those factors that we all know about as to why biologicals are getting attention, I want to talk a little bit about the technology advances that are taking place that are also getting the attention of the major agrochemical companies, the seed companies, and how biologicals can be evaluated more efficiently and be developed into superior solutions. So first, some of you have probably already seen this chart from the NIH, and it talks about the precipitous drop in the cost of a full genome sequence for humans. 2001, it took $100 million to do the first genome sequence. Today, even though it's not on here, 2015, I believe the estimates are now $1,000. The importance of this is that this is greatly facilitating our understanding of live systems, and it's becoming much more cost-effective for us to look through a wide array, hundreds and thousands of microbes in a cost-effective manner to better understand how and why they work. Next, a very important thing to the world that I live in now at BioConsortia is the understanding of the microbiome. Extremely reputable scientists are increasingly becoming very familiar with and comfortable that the microbiome is a big part of healthy human, animal, and plant systems. This is what, as all of those industries and sectors enhance their learning, those things are going to be more and more available for more effective discovery and development programs to ultimately deliver more consistent biological solutions. So now I want to get on my soapbox and talk about what we need to do better. And part of us, all of us, including companies like BioConsortia, need to do a better job of finding a way to integrate with current standard agronomic practices. And those current standard agronomic practices include chemistry. Are we compatible with chemistry that's in the tank, being sprayed at the same time, on the seed, that is trusted and valued? Are we delivering stable solutions? Stable in the package, in ag warehouse conditions? Are they also stable on the seed? Again, compatible and stable on the seed in the presence with chemistry. And finally, making sure that our biological solutions have the right use rate. Is there enough space on the seed for your biological solution to coexist with chemistry? So historically, in general, I think the biological industry has done a poor job in this area. I think we're improving, but we still have a long ways to go. Second, I'm going to give a lot of credit to the program that Monsanto has put in place and talk a little bit about why they're doing what they're doing. I think Brad touched on some of this. But field testing, extensive field testing, it is critical with the biological. Having been, again, on both sides of the table, fighting to develop products and then evaluating products, we often fall short in our understanding of our biological solutions from a field evaluation standpoint. Because often enough, the biological solutions have more of a potential of differing effects when it comes to genetics, as well as in varying different environmental factors. Particularly if you have a live microbe, you have a fermentation process. And anybody who knows fermentation know that temperature, pH, nutrients all drive your performance in that fermentation. So until you go out and you look across a great degree of environmental factors, you might have you might find you have an inconsistent product. So, or you have a niche product. But the key is to understand where your product works and probably more importantly, where your product doesn't work. So just to touch on the question of, well, how is, here I'm beating up the industry as a whole, how we've historically been, what is BioConsortia doing about that? Well, one way is we have a very unique discovery process where we allow the plant to do the heavy lifting, for the plant to do the screening for us. Well, what is on that seed? When we plant the seed in very diverse soils, what is on that seed? We're starting with chemistry-treated seeds. We are looking for things to be compatible with chemistry, and we're looking for things to add value on top of that standard agronomic practice. We're not trying to fight and displace what is there. We're trying to enhance what is there. Lastly, I just want to leave you with a couple great examples and talk about why they've been very successful. One is Votivo. That's a root colonizing bacillus. It's a very stable bacteria. It colonizes a root to provide extended benefit into the growing season, something that a traditional chemistry seed treatment can't bring. And probably you could argue that the greatest part of its success was that it was partnered. It was co-formulated with one of the market-leading insecticides. So I think this is why Bayer bought AgriQuest. This is why Monsanto went into a collaboration with Novozymes, why Syngenta acquired Pasturia, why BASF acquired Becker Underwood. It's how we integrate these solutions to deliver something that can't be delivered with the other. And lastly, just to real quickly highlight another great example of a biological that found its way to integrate with chemistry. You don't get to 50 plus million acres, annually without working in conjunction with chemistry. So with that, I think I'll turn it over to a brief discussion and questions.
Excellent. Come on up, guys. So let's have a few questions. I know we're almost out of time, but I'd love to kind of open it up for any questions from the audience. Back here. So the question is to the concerns that regulatory agencies might have, particularly as you combine biologicals with chemicals. Anyone take a crack at that?
I can maybe start to give an answer. I mean, I'm not familiar enough probably with the US system. In the European one, what you will have to do first is to ensure that the two actives are registered independently. And then when you mix the two actives together, the challenge, as I tried to explain earlier, will be to demonstrate the added value of the two compounds together versus the individual product. And that's more and more the case. And I think that's not a secret, but we have to, let's say, reevaluate the pesticide and the mixtures every 7 years in Europe. And now the authorities are challenging mixtures which were granted a registration 7 years ago. So the justification in some areas of the world will be critical for that.
Any other thoughts on this issue of integration, Brad?
I could take a run at it, sure. And I thought you did a nice job actually in your opening comments. Just the overall, I think the regulatory agencies around the world want us to be successful, and they've been extremely open and receptive to listening to and reviewing our data. So I think that we're going about it the right way as an industry. I know at Monsanto we've actually added a significant number of resources to our regulatory team around the world to make sure that they've got the tools, the knowledge, not just the what, but the how, and to be able to answer all of those things for our regulatory teams. We have, again, with Novozymes also expertise that helps us to interact with the authorities. So I feel like we talked about the relative age of this category compared to others. I think our regulatory brethren certainly see it as new as well, and to the extent that we can be very transparent and open with them about what we know and how we work with them as an industry, it's only going to make our regulatory interactions around the world better off.
So how do you deal with the fact that if I have a bug that makes a nutrient more available, that's a fertilizer, and if I have a bug that attacks another bug, that's a pesticide? I mean, how do you deal with that from a regulatory standpoint? Because at least my sense is we have a problem because regulators they want everything in a bucket, right? And the bug doesn't get up in a bucket, right? It does what it does.
Yeah.
And it has a lot of different effects, and regulators have struggled to kind of keep up with what does this mean? How do we regulate it?
Yep. Go ahead, Joe.
I was going to say, I think they continue to struggle with it because a microbe is a biofactory, not of just one thing. And often enough, a microbe brings multiple modes of action. And so traditionally, I think, again, it depends on the market. They're choosing to regulate it based on what claims you're seeking to make. And I think it's an evolving time on this very point.
Yeah, and I think the complexities associated with what you're inducing, in some instances, what a microbial may do to the plant in terms of enhancing genetics only adds to the complexity. So in fact, I mean, yeah, you're looking at how can you enhance nutrient uptake by root architecture, what are potential antifungal components that provide protection, and then ultimately, can you induce the plant to enhance its own protection devices? I think it's the claim. Initially, it's going to be the claim. And I think that moving forward, I think it's going to become even more complex. And particularly my concern is, as we move forward in terms of the regulatory process globally, is what are the implications associated with indigenous microbials? And how does that become part of the mix in terms of being able to regulate or establish a registered product in other countries, and specifically in South America? So
It's a very slippery slope.
I think a trend that we do see that the more, let's say, R&D companies, startups are in this space, and the more the regulatory requirements are fine-tuned. You know, technology is ahead of regulators, as very often. You know, it's like a safety with cars or plates. You know, and what we see is that we won't go backward. I think the requirements will be more and more stringent. It's not only true in the US or in Europe. In Chile, for example, it takes at least now 3 years to have a registration, right, which used to be simpler in the past. So let's be lucid about that trend.
Excellent. Other questions? Yes.
One theme that keeps on reappearing today is the idea that technology is at such a pace it outstrips that of regulators, of consumers, of a lot of the rest of the world. How do we fix that?
I'll take a run. It's a great question. Yeah, the question was you tell me if I paraphrase this correctly, the technification in agriculture and in this category in general is moving at such a rapid pace. How do we ensure that regulators and consumer, multiple constituencies and stakeholders keep up? Is that good? Yeah. You said it more eloquently. But I think it's something we manage at Monsanto on a daily basis. Stephanie Reganin here sitting in the front, her team is a part of really trying to get that message out to certain stakeholders. And we're up here, our eyes are distracted a bit because we're watching the blogs that are real time, and it's so cool to see this coming out. But this mobile technology trend and Twitter and Facebook and pushing articles out that really help educate friends of friends. And we all go to parties and we all have people that we hang out with socially, and education is a really big deal. So I think really helping to educate them on the opportunities that exist, how these products work, is going to be paramount. So leveraging technology and outreach.
yeah, and not talking to ourselves. I mean, that's what wears me out. I mean, it's great to, you can certainly create momentum if you're talking to the same people that are, utilizing or benefit from the technology. But I think the dialogue has to be, you have to be willing to engage in dialogue that can be unpleasant at times. Because I think at the end of the day, it's going to become a very, as Brad indicated early on, as we talked about, we have a lot of people to feed. And if you take away the current crop protection tools that we have would require the tillable acreage of South America, what we currently can do with just North America. So if you're adding more, you've got to be willing to stand up and make a case for the technology, but also understanding the cultural aspects and ramifications of what you're talking about, where it goes. And that's one thing that I don't think that we've done a very good job of that. And so in that respect, I think that it's a combative dialogue that hopefully will have a positive end. And I think that understanding the ramifications of what we do and how we can move forward together. And there you go, there you have it.
One more, Kirk.
Thank you. Are biologics going to follow the same path as the seed industry, meaning from a path to market? So are you going to require, the same way you need certain types of germplasm for Brazil, Argentina, that's different in the United States or Europe? Are biologicals the same? Or is it going to be, hey, the same bacteria that makes corn grow really well in grass is going to be the same globally? And so how do you handle that versus
I think it's a great question. We have a process that is relatively efficient to be able to customize that if you need it. But I can just speak to some examples for us. our company started in New Zealand, and this past year, yes, it was a small set of field trials, but we showed leads that came out of New Zealand, applied in U.S. Standard growing conditions, and we showed them perform just as good, if not better, than they did in New Zealand. So I think we have a lot to learn in that respect. There's potential to be able to customize it with biologicals. It's a lot easier to do that because the efficiencies in biologicals from a registration and a safety standpoint. So I think the benefit is that we have the freedom to do that, and it's efficient to do that if it's needed.
Yeah, I think that what's interesting and not often talked about is that when you put, because I think we're all very much, a lot of us are very much in tune with chemical crop protection and a ubiquitous product on a lot of acres and a lot of crops. But clearly when you put a biological system on another biological system in another environment, things are going to happen. And so I think that you have to understand what those are. Yeah, I think there are going to be differences. There are going to be unique opportunities to be able to leverage biologics. And so that's an important area that we, and New Leaf looks at in terms of leveraging bioinformatics, so that we have the capability, as Joe said early on, the technology is available. We have to use the technology to better fit and to address those unique environments and be willing to say, yes, this doesn't work all the time in that environment, but we do have something that does.
We talked about trials. I think everybody in the panel talked about the importance of trials and establishing credibility for this category so that farmers have an agronomic and economic rationale that they can pin their input decision to. One of the things we've done at Monsanto is with the acquisition of the Climate Corp, we're integrating Climate Pro into all of our field trial efforts. That will allow us to access multiple levels of data and really understand not just the what, the output of yield, we saw it, and take it somewhere else and see the output of yield, but why? What's going on? So that empiric approach, I love it. I'm sure that's out there. We're going to find it, right? We'll find it together. But there's certainly going to be learnings of individual technologies that fit in a certain soil type, certain pH, certain GDUs, and you've got to have the data analytics to pull all of that together to make the right, to make intelligent genus and species selections for your next testing campaign. So I think we have a lot to learn in that regard, but we'll make progress, and I don't think it'll take us an incredibly long time.
Excellent. So I'm sorry we're out of time, but please join me in thanking the panel for spending the time. Thanks, guys.
Thank you.